It's groovy DNA. I'm so sorry. I think that's what the book called it. I literally think it was a subsection like titled that. So B form, beta DNA, original DNA, canonical DNA is another name I might use. Okay, sort of the OG structure as described by Watson and Crick on the basis of some other people's X-ray diffraction data. Okay, but we have some sort of key features. So this is sort of a top-down view if I'm looking at the helix. I have a convex edge, which will form part of the major groove. I have the concave side. I have sort of my base pair here. This concavity is sort of a result, mostly of the glycosidic bond. It's details I don't need you to memorize, right? But some key visual features overall. Base pairs are oriented perpendicular to the axis. The asymmetry, like I said, in sort of the glycosidic bond is sort of what gives the concavity. It's that CN sugar to base bond that's leading to this shape. Typical form is going to be right-handed helix. And our two grooves, the minor and major groove, are of unequal size. Kind of makes sense based on naming. And so those sort of the top four like obvious features of sort of this, maybe from a more 3D overall structure, what you would observe. Okay? Getting away from sort of the individual interactions there. So the major groove is roughly twice the size as the minor groove in B-form DNA, and so you can sort of see this based on the scale. Okay, this indication here, and so these versions, sort of these spherical ones, or even this is sort of, they're hard for me to personally visualize. It's always hard for me to decide what stick model I prefer on any of these. But what you can see here is this is sort of across the entire major groove and across this sort of bigger gap here is about 15 angstroms, and then in the minor groove, closer to 7. So roughly half, right? The major groove is providing greater access for the things that want to bind to DNA, right? Most stuff is going to bind in the major groove, not only, one, because it is bigger, right? That's part one. And then sort of the other reason it might bind there is part two. But this could be proteins that need to bind, enzymes that need to bind, drugs that want to bind. When it's time to transcribe DNA, right, or replicate DNA, we have these whole complexes that need to attach. Okay. So in addition to size alone, what other factor might contribute to why things want to bind in the major group? Or I should say, what other types of things dictate where anything wants to bind? Yes? Perfect. The atoms that are available in the base pairs there, right? And those atoms might be the kind that can make hydrogen bonds, might be the kind that can make some other type of polar interaction. Maybe it's methyl is hydrophobic, so maybe it wants to make a hydrophobic interaction. So it's all about sort of the pattern of what's there. So non-covalent interactions are the other reason why anything binds to any particular site, is sort of recognizing a pattern of bonds that it can make. So complementary patterns at the contact site between whatever the molecule is and the DNA strand. This is another thing where if I want you to use this, I'll give you this figure. I'm going to ask you to memorize which types, H bond acceptors or donors, or hydrogen atoms. So we have all our labels here, right? We have donors are shown in red, sort of red molecules. We have hydrogen bond acceptors shown in blue. We have a hydrophobic group. Like I said, a methyl group is going to be hydrophobic. And then again, something that is not sort of like a hydrophobic methyl, but just as a nonpolar, just plain old hydrogen atom not attached to an electronegative atom is shown in gray. And I don't need you to know that for a GC pair or a CG pair, depending on how you're orienting it, what ends up on the major side, the minor side, like this memorization. What I would want you to maybe know is if I, showed you a structure you hadn't seen before, could you circle something that could be an H bond donor acceptor? Like if I showed you the protein side, the molecule, the little molecule that's coming in, could ask you where might it want to pick out of this list I gave you. Yeah. Which ones are?
The donor is red.
Yep, they are. Good call. Donor is blue here, red is acceptor, and I have my color coding flipped. But again, I could give you this. Then I could show you some little molecule, and you could see that it has H-bond donors and receptors on it. And I said, it's really big. It's 16 angstroms. It has to get into the major groove. What sequence of base pairs would it need to get? And you'd go look at the major groove and go like, oh, it only has donors. It wants acceptors. Let's maybe this pair, maybe this pair. You could argue something to me, right? So in order for replication, transcription to occur, protein machinery, these big complexes, RNA polymerase and whatnot, have to be able to recognize a specific region of DNA in order to bind. And that's based exclusively on whatever this pattern of sites is. Okay? And again, they need to, this from the outside is kind of how the book calls it. And it again is, this pattern needs to be in the minor and major groove so that it'll be able to bind there. I think I may have taken, no, I took the slides out. Okay, so back to this. Again, since we are talking about if something's going to come in and interact, right, at the major or minor groove, it's going to make some H bonds, potentially maybe a hydrophobic interaction with DNA. So we're talking about, in that case, intermolecular energy and some summation of these different things it could interact with. Okay? Kind of ties it back in. DNA is deformable, so we have our idealized beta form, okay? That's not real life. In reality, DNA is actually quite flexible. A way to think about it is that B form is sort of an average structure. If you have a whole bunch of DNA helices, they're sort of fluctuating, and kind of on average is the structure it'll be. Why do we have fluctuations in size? Why do we have deformability? And sort of two things I'm looking for.
Because van der Waals interactions are really transient.
They are transient. Now we're saying that they're pretty stable for our, those base stacking ones, but indeed, when they're transient, what was the thing that can kind of disrupt them?
Water.
Water's not going to disrupt the base stacking van der Waals.
Is it like based on the environment around it?
Yes, it's all in the environment. But what was specifically the thing that I said, like, this is lower energy than van der Waals and it messes them up? There's a type of energy, and I think fluctuation maybe helps with that. Kinetic energy, thermal energy, love it, right? So fluctuations in sort of this overall shape and what it looks like at any given instant is binding, what's binding to it, and also thermal energy driving collisions with other molecules, including water, including biomolecules. Again, examples I won't, distomycin, I don't need you to know any particular drug that's binding, but what's being shown in this figure is that you can come in and sort of elongate the minor groove to get in there, like DNA is very flexible. Proteins also very flexible. And when we talk about molecular recognition, we'll talk about proteins basically being able to almost like breathe, like open and close to let things bind. Okay. So again, original structure, but DNA is deformable, and we have two grooves of unequal size, and more importantly, they have sort of a different number and pattern of recognition sites. Okay. This is a nice table that I want you to have knowledge of its existence. And you would feel comfortable using a number off this table, but again, don't memorize exactly the helix pitch between each term in angstroms, not important to me. Relative scale is what I care about. Okay, nice summary, don't memorize, at least not for me. And again, this talks about alpha helices and Z form, like A form and Z form instead of this B form. Again, you don't need to memorize the differences. If I wanted you to say anything about them, I'd point you right to the table. Okay, RNA structure, this super briefly. The hydroxyl on the C2 presents a steric clash with the phosphate backbone such that it can't assume B form helices. So rather, when you do have double-stranded RNA, it's more likely to assume A. And again, you can just look at that table to get ideas about some of the differences in angles and size. But RNA is especially versatile in its structure and its function. It's not always it's definitely not always a double-stranded A-form helix. It can be in many forms, and because of that, it's super functional. right? So we have mRNA, what does that stand for? The tRNA, what does that stand for? And so this is involved in translation of proteins, right? So we have, you know, and I don't think, we're not gonna get to it, like I said, I wasn't really gonna go into the protein stuff, so actually looking at how translational like RNA looks kind of like the clover leaf thing, right? It has a very unique shape that's related to its function. And whether it's going to have some sort of non-standard base pairing loop or not will be based on what it does. But it's important to remember that it has the ability for those things where DNA really doesn't. Okay? And if I asked you like, oh, I want to make some synthetic RNA and I want to use it for something and I need it in some weird shape. Let's say I ask you, I want to make something synthetic out of nucleotides. Would you use DNA or RNA? I'd hope you'd say RNA because I don't think DNA is going to be flexible enough to get into anything else, right? I need that extra flex of RNA. Okay. There is still base stacking in RNA. Okay, so RNA can have, sort of depending on how it's decided to complex, whether it's sort of looped back on itself, or it can have some regions where it is forming some of these van der Waals. Okay. Okay. A couple of illustrations of how RNA might do this, right? It's single strand and it comes back and folds on itself. Again, base stacking is ultimately sort of the driver for the stability of these helices. These are old slides, I should fix some of these words. Because I want you to remember that the phosphates help dictate the helical structure, with van der Waals in base stacking being about the stability. The bases are still flat, yes, yes. And then here's just an example, a little zoomed in picture of how RNA would, again, have alignment of the bases in this helical direction.